Okay, folks, welcome to the afternoon. This afternoon will be a lot about plants, or at least some stuff about plants, which is pretty exciting. Hopefully you didn't miss the JGI World Showcase where I got to talk about plants already. Our keynote speaker this afternoon is going to be Michael Purugganan. Michael's a professor at New York University. He's a professor of biology, and he's a dean for science, Faculty of Arts and Sciences. So Michael got his PhD at University of Georgia, which is weird because there must be a lot of Georgia people in here right now. Oh, come on, there's got to be a few more. Did his postdoc work, which is what I'll mention here at the University of California in San Diego. And one of the things that Michael worked on early in his career were these things called MADS genes. And these are genes that are involved in flower development that have this very long and interesting evolutionary path where they've been duplicated over time and then taken on different kinds of functions in floral development. He's well known for that, and also for looking at domestication events and looking at the domestication of how we convert a sort of wild species into a domesticated crop species, and what kinds of impact does that have on the genome, and how over evolutionary time does selection happen to these plants that are in the wild. What I wanted to say was that Michael, you got to come up. Come on up. Come on, it's super important. There's a very funny thing on Michael's webpage, which says that he's interested in slime molds. And for those of you who are not plant people, there must be some people here in the audience who are interested in slime molds.
Big P? Anybody working on Big P? Woo!
Oh boy.
I stopped working on it already, unfortunately.
Oh, that's too bad. But I want to welcome Michael, talk to us about adaptation in rice development. So.
Yeah, thank you. And thank you very much. And thank you for unearthing that ancient history with the MADS genes. And interestingly enough, at the end, we will talk about a MADS gene, which, everything comes back full circle. So many of you probably know me. How does this work? Point this way? This way? Next? Yeah. Okay. And thank you, by the way, Jeremy and the organizers for inviting me. It's really wonderful to be here. As Jeremy pointed out, many of you may know about our recent work on looking at domestication of crop species and crop evolution. But that's not what I'm really going to talk about today. I'm going to talk about another area of interest in my lab that we've been doing for the last few years. And that is we want to try to understand how selection acts on crop genomes. And here specifically, about a few years ago, we decided to launch two fairly large projects to try to understand, especially the evolution of gene expression in the rice genome, and in so doing, hopefully open it up to looking at selection in a variety of different genomes, plants or animals. And so the reason for this is that we kind of understand as biologists that the levels of expression of a gene are of course vital to the translation of genotypic variation to phenotypic variation. The ability to regulate that gene's expression in time and space, and the levels of gene expression. So there's really an important feature here that we have to understand, and how is selection acting on both the regulatory elements that may underlie the expression of those genes and the levels of gene expression itself. So today I'm going to talk to you about two different projects. And these are the two postdocs who have taken on these projects several years ago, and they're now wrapping these up, and we're about to send out the papers that describe this. One is from Zoe Jolie Lopez, who undertook the task of basically making a map of fitness consequence across the entire rice genome. And what is that? And you'll see later on what that is. And then Niels Groen, who was looking at trying to understand how selection acts on the expression levels or the levels of transcript in the rice genome. So let's jump immediately into these two areas. So what's a rice fitness consequence map? The idea for this came about from Adam Sipple at Cold Spring Harbor several years ago. And if you think about mutations and polymorphisms in the rice genome, or any mutation that arises in any genome, you know that there's a distribution of fitness consequences. So many of the mutations that arise are deleterious and will be selected against. Many will be neutral or nearly neutral and will drift around the genome in the population. And several will be in a class that may be adaptive. And these are the ones that we are kind of interested in thinking about evolution in the long term. And thinking about one of the challenges in biology is if I gave you a map of the polymorphism and divergence across a genome, can you say what is the fitness consequence of each of those polymorphisms, right? So can you point and say, this is neutral, this is nearly neutral, this is adaptive, this is deleterious, and so on. And having such a map would be of tremendous importance, right? If you were in human genetics, it would help you try to understand how mutations, what polymorphisms underlie genetic diseases. And in the agricultural context, in breeding, you would understand which mutations or polymorphisms you may want to remove and which you may want to bring onto the next generation. Now, there are many ways to try to get these fitness consequences. Some of them are use evolutionary conservation across different species. And that really works for coding regions or conserve non-coding sequences. But for the vast majority of a eukaryotic genome, they're non-coding DNA for which annotation can be sparse. We don't know the nature of the function of those elements, much less what are the fitness consequences associated with mutations in those regions. And this is actually a big kind of black box or a dark area that we're trying to uncover. So how do we do this? As I said, Adam Sippel came up with this method several years ago when he was, I think he was at Cornell then, and then he just moved to Cold Spring Harbor. And one of the issues when looking at, say, regulatory sequences in an uncoding region, and you're trying to use population genetic techniques to try to infer selection acting on those regulatory sequences, is the fact that these regulatory sequences are in general fairly short. So from the population genetic standpoint, there's not a lot of information you can glean from these short sequences. And so the insight that Adam came up with, what he said is, well, for each particular regulatory element, we might not have enough information, but if we have a class of elements that behave similarly across the genome, and we treat them like they were one class of elements and use those together to give us the signal of selection, we may be able to infer the strength and pattern of selection across these class of elements. And so he called this insight the inference of natural selection from interspecies genomically coherent elements. It was a method to try to infer selection on even short regulatory elements by integrating them across the genome. And what this data, what this shows, and unfortunately I don't think I've got a, oh, I do have a pointer. Is this, oh. That's fairly weak. Anyway, what the insight method does is basically it solves this equation for looking at, it takes polymorphism and divergence data within species and between species, and it calculates a variety of evolutionary genetic parameters. And the one that's most interesting to us is the one on top there called rho, which is in a given element, a regulatory element or sequence, whatever you've got, what is rho? What is the fraction of sites under selection in that element? So if it's 0, that entire sequence is evolving neutrally. There's no fitness consequence associated with mutations in those sites. If it's 1, every mutation in that site actually has an impact on fitness, either positive or negative. And so he had developed this method called InSight. He published it, several papers, about 2013, 2014. And what we did in the lab was we took InSight, which he had developed really primarily looking at the human genome, and we developed what was called green InSight. And you can't see, unfortunately, this is a huge pipeline. We had to adapt the InSight method that Adam did to work in plants and to take into account the peculiarities of plant genomes, and we can talk about what those peculiarities are later on. And so what we decided to do was to try to apply insight into plant system, in this case in the Rhizis sativa. So what we did is we took population data from about 180 lines that we sequenced in my lab and about 1,400 from the 3K Genome Project, as well as 12 outgroup species from Rhiza, some of which was provided by Rod Wing in his OMAP project, some of which we actually also sequenced ourselves with some quick de novo assembly. So we used this as a population genomic data set. And we applied this InSight method to a variety of broad categories across the rice genome. And here we compare what it's looking like for sativa versus, we did the same thing for Arabidopsis thaliana, and we also compared it to Adam's results in humans. And you can see that in general, the results are concordant across these three species, and they're concordant, and we can compare it. For example, the neutral sites have very low row value, conserved non-coding sequences and coding sequences, conserved non-coding sequences and coding sequences have fairly high row values, and then the rest have intermediate levels of row values. And the levels across the different species are comparable to each other. So the Green Insight system was working in our lab. So what we decided was to use this in an iteration of what Adam did, which was to get the Insight model and to try to use it and map fitness consequence scores, or these row values or selection values, across the entire genome. And he had written a paper in Nature Genetics in 2015, which he called the fitness consequence, a way to infer what he called the fitness consequence map of rice. And the way this is done is kind of really interesting. There are really 4 steps in developing the fitness consequence map of a species. The first is you get functional genomic data. Remember what Insight does. It says, let's look at elements across the genome and put together elements across the genome that have very similar features. So what we do is we collect a large number of functional genomic data and epigenomic data across the genome to allow us to classify across the genome, especially in the non-coding sequences, sequence elements that behave the same, that have the same fingerprint. So we cluster that in step 2. We get the functional genomic data. We cluster these elements across the genome by these functional genomic fingerprints. We apply Insight. We get the row scores, and then we map them back to the genome. So it's a 4-step procedure. It seems relatively trivial. This took us 4 years to do. And in order to do this, as you'll see, my postdoc, Zoe, essentially was a one-woman rice ENCODE project. In order to do this. So the first thing to do this, step 1, let's generate this functional genomic data. And we sat around and we looked at Arabidopsis data and others to try to understand which set of functional genomic or epigenomic features would make sense in trying to classify non-coding elements across the rice genome. And so we had 11 types of functional genomic information. We did total RNA sequencing, we did small RNA sequencing, we did attack sequencing for chromatin accessibility, we did DNA methylation, we did 6 histone marks for chip sequencing, and we did precision run-on sequencing, which was just developed last year, which allows you to map active polymerases in the genome. And this, by the way, for those of you who want to do this, this is a monster protocol. It's 175 steps. It takes a week to do. But Zoe's, she got it working the first time. And then she replicated it next week. She got it working again. So we're kind of really proud of being able to get that to work in the lab. And so what you get is now you've got all of these fingerprints, you've got all of these features across the rice genome. Now we focus on leaf tissue. So obviously if you did different tissues, you would have a different set of fingerprints, but we focused on leaf tissue in this, and we're able to get a wide variety of different fingerprints. Now the next thing is to get this functional genomic data, and you aggregate them into classes of elements that share similarities among these functional genomic fingerprints. And in fact, just by looking at it, you can start to see different things. You can see promoter regions, areas of nascent transcription or even transposons. You can start to classify them into broad categories. The way we did that is we use both Chrome HMM, the hidden Markov model for looking at chromatin states, and intersecting this with a variety of different genomic annotations, including some of the genomic data we have, like stable RNA or coding regions and exonic regions. Essentially, when we did this on our data set, you can get about 640 possible classes, but only 246 include elements that together make up at least 20 kb of the genome. So the others are essentially empty sets. They're very, very unique, or they only are a really small fraction of the genome. These 246 final genome classes that we've managed to get in the rice genome have at least 20 kb coverage or more across the rice genome. 246 classes is distributed about 4 million blocks. So the genome is partitioned in these blocks of 10 to 40, on average 10 to 40 nucleotides. So each of those classes have an element between 10 to 40, but some of them can be fairly large. And this just shows you some of the class blocks that we have in our data. So, okay, so now we have this functional genomic information. We've managed to stratify the rice genome into these different genome classes based on genomic and epigenomic data. Now let's use Insight, and let's use the population genomic data and the between species data to calculate this value rho, this fitness consequence score, which is the fraction of sites under selection in each of those genomes, and then assign that to the different genome locations. And of course, we are able to do that. So this just shows you the visualization of the genome browser we're going. The one in the box shows you where you get the usefulness of the sequences. Unfortunately, since I can point, you'll have to look at some of this. In some of this, you'll see regions of attack-seq that's open chromatin underlying a gene. So the gene track, you can see that there's a gene there. And if you look down at the last track, which is the fitness consequence score, you'll see that the rho is fairly high. These are coding sequences. It's showing you coding sequences. They have a high rho, meaning that a lot of mutations in those regions are going to have a consequence to fitness. But where this is useful is if you look at the area under the box here. And what this is this is an open chromatin region. It's got fairly open chromatin based on attack-seq. If you look at the genome annotation, there's nothing there. It's non-coding region. There's nothing annotated there. But if you look down at the fitness consequence track, it has a high rho value, suggesting that it's under a strong selection. So this is where this method can be useful in genome annotation, because now you're able to go through stretches where you don't have any other form of information, but now you know that selection, high selection, is happening in those regions. The distribution of rho across these classes along the genome is bimodal. About 10%, this is just by classes. And it says that about, several of the classes have a rho of about greater than 0.5, approximately 0.75. These are the high rho classes. In terms of genome coverage, they only represent 10% of the genome. So only 10% of the genome is in this high rho category. The majority of the genome is in the low rho category, rho of less than 0.4. We're still trying to play with this because we're a little bit concerned that that first mode in that bimodal distribution has a rho, mean rho of about 0.25. And we think it's a little bit high, and we think we know what's going on there. So we're playing around with a few things to try to see if that holds out. And if you look on the right, you'll see how rho changes, how the coverage loss of genome changes with rho values, comparing the human genome and the rice genome. In the human genome, pretty much everything is low rho until you hit a rho of about 0.1, and then it shoots up. Everybody is about high of 0.1 and above. In the rice genome, it's more gradual. Elements have varying levels of selection in different elements, and the coverage across the rice genome increases gradually with rho values. So that's something that's interesting. Just to also check ourselves, we would expect that if you have a high rho value, that is, you are a sequence that has a high fitness consequence score, that you're going to have more rare SNPs in the population genomic data set. By a variety of different methods, we've shown that that's true. There's a shift to rare variants under high rho, which tells us that the way we're doing this seems to be working well. Just to show you some of the things we're looking. For example, we're looking at different types of rho values. Here's the intergenic region. You can look at the rho value in general. This class of intergenic region, which has no open chromatin, no coding sequences, obviously no active polymerase. It's got very low rho values, less than 0.1. But look at these transposable elements. This area that has transposable elements also have rho values, but then you're beginning to see looking at different genome annotations and what selection is acting on them. And by putting together both the rho values and the different genomic and epigenomic tracks, we're able to see characteristics, genome characteristics of different classes of elements. This is coding sequence, again, high rho value. If you look at the dash box on the left, you'll see high enrichment of coding sequence and exons in these classes, and the rho values are greater than 0.8. Again, something that we would expect. This is an interesting region. These are intergenic regions with a high rho value, okay? These are intergenic regions with a high rho value. And then here's another one, which are intergenic regions with kind of a low to moderate rho value, but this has open chromatin and also active polymerase. So you're beginning to start to see a stratification, as I said, of the rice genome into these different classes and the action of selection on these different classes of elements. The intergenic classes with high rho, we're able to show that these classes show a high density upstream of transcription factor genes. It also is in high density in regions that have a high fold change between tissues, suggesting they're developmentally regulated. These are regions of the genome, as I said, these are intergenic, but have high rho values. This class, we're still not quite sure what to make of it. These have low rho values, but what they have is a high pro-seq value. So there's a lot of active polymerase in these classes. And what we've shown is that there's a weak but positive correlation of the presence of these classes with expression of nearby genes. And you can see the numbers there of the correlation. Even though the correlations are low, they're quite significant. And one of the features of this is that if you look at the pro-seq data, you're seeing bidirectional transcription from the active polymerase, as shown in the box plot here. Both the plus and minus strand show evidence of transcription. And that's interesting because this is characteristics in animals of enhancer RNAs, which are associated with enhancer sequences. So we actually have about 1,000 elements in the genome, 1,000 sequences in I think several classes that showed these bidirectional transcription associated with enhancer RNAs. And when we do an algorithm that detects for these enhancer regions based on the pro-seq bidirectional transcription, you can see that all of these low rho areas with high pro-seq also have an enrichment for bidirectional transcription associated with enhancer elements. So I'm just really going through all of these just to show you some of the data that we're able to obtain from there. And finally, these candidate enhancers that I show you, we're able to find that if you look at these candidate enhancer regions, remember, we don't have any other information about them except that they have bidirectional transcription from active polymerases. There's an enhancement in open chromatin around them, and there's also asymmetrical localization of this histone mark that's associated with transcription. So we're winding this up. We're just putting we're trying to figure some of the behavior of the low rho classes and also cleaning this up and writing up the paper. In the end, what we're going to have is we're going to have a genome browser, which has all of our functional genomic data, as well as this fitness consequence track, and we hope that it'll be useful for the general community. This is going to go online as a resource from our lab in the summer of 2019. We'll have an insight in FitCon's website. It'll be a mirror of the UCSC genome browser. You'll be able to plug in your favorite gene or area of the genome and find what the fitness consequence score, what the track looks like, what the genomic and epigenomic marks look like. So stay tuned for that. So in summary here, what we've developed, as I said, is a map of the rice genome where we can now attach selection strengths across different classes that are found across that genome. And more importantly, we can do this even in non-coding regions of the genome where we have a sparsity or a paucity of functional information. And where this is going to be useful are in two things. One is we think it will be useful, hopefully, for breeders who are looking and trying to figure out if they have a GWAS or a QTL map, and they're trying to understand where there might be regions in their genome region that could have mutations with a phenotypic consequence. This might give them some suggestions if they're able to localize it quietly, because we're able to show different fractions of the genome with different rho classes. And the second, as I've shown you, is that there's a back and forth between the functional genomic and epigenomic data and these rho scores to try to understand these functional genome annotations, these functional genomic marks, what they're telling us, what are these genome element classes. Hopefully it might be a way to help continue to annotate other genomes as well. So that's the first project that Zoe Jolie Lopez did, looking at developing the fitness consequence map of the rice genome, and we're going to continue that project, basically looking at other tissues as well and other species. That's kind of part 2 of that project. The next one is a project that I've actually wanted to do for 15 years. It was a very expensive project, but finally the technology caught up, and now we can do it without as much cost as we thought 10, 15 years ago. And the idea here is, can we measure the strength of selection on gene expression? Okay? So if you, again, think about what's going on in the expression of traits that are associated with fitness, in this case, the number of seed of rice, there's, of course, genetic variation. And that's leading to phenotypic variation, which leads to different fitness outcomes. Is this plant going to live or die? Is it going to produce a lot of seed or not a lot of seed? And we know that, as I'll show you, you can measure the strength of selection on the trait itself. That is the phenotypic trait, such as height, chlorophyll content, whatever you want to do. But one thing that always intrigued me was, can you actually measure the contribution of the levels of the transcript levels, the levels of gene expression to fitness, and thereby measuring the strength of selection? And the way to do this has been known by evolutionary ecologists for more than 35 years now. And it has to do with multivariate selection theory. Basically, in multivariate selection theory, the response to selection of a given trait is just a product of the genetic variance covariance matrix, that is the amount of genetic variation you've got in the population, the phenotypic variance covariance matrix, that is the amount of phenotypic variation, and this value S, which are the selection differentials, that is the strength of selection. So how much you can move a trait to the next generation has to do with how much genetic and phenotypic variation there is in that trait and how strong is your selection. So that's simple. That's classic quantitative genetics. And the inverse of the phenotypic matrix and the selection differential actually together make up what's called the selection gradient. Now, Landy and Arnold had this paper in 1983, which really was a landmark paper. There's about 5,000 citations of this paper. And basically said is prior to this, when evolutionary biologists such as myself talked about selection, frankly, we didn't quite know how to measure it. And what they did was they came up a way to measure it. And so instead of us just kind of guessing whether this trait is under positive selection or negative selection, they said, let's measure it, and we can see what is the selection acting on that trait. And what they said is if you have fitness W is really caused by two things. By linear selection, that is directional selection, positive or negative selection, acting on a particular trait phenotype, and quadratic selection, which tells you stabilizing selection or disruptive selection, acting on that trait phenotype. What this allows you to do is if you have a particular trait, such as, let's say, plant height, and you want to know the strength of directional selection or linear selection and the strength of quadratic selection, if you had fitness data and that trait data, you can actually estimate those different parameters. And in fact, that's what evolutionary ecologists have been doing since this paper came out. By the way, this is just the different types of examples. If beta, the absolute value of beta, or the selection gradient, is greater than 0, you actually have directional selection. If your phenotype is increasing, your fitness is going to increase. Or if your phenotype is decreasing, your fitness will decrease as well. There's a direct correlation. And then a gamma greater than 0 gives you stabilizing selection. Gamma greater than, less than 0 gives you stabilizing selection. Greater than 0, disruptive selection. Actually, in practice, the quadratic selection term doesn't turn out to be that strong in most wild populations. But evolutionary ecologists have taken this data set and have measured everything that they can in a large number of species. There are thousands and thousands of measurements of the strength of selection in animals and plants. And they've been, and this is from almost 20 years ago, where these are just papers that strength and temporal directional selection, strength of phenotypic selection. These authors, Hopi Hoekstra, Joel Kingsolver, have taken all of these data that their colleagues have taken and they put them together and tried to understand what are all of these studies telling us about the type and strength of selection acting in natural populations. And what they come up with is this. They come up with essentially selection is relatively weak. The distribution of selection coefficients is exponential with a long tail. And in general, most selection is, the selection gradient is less than 0.25. And this thing has held out. It doesn't matter whether your trait is morphology or life history or animals or plants, the distribution looks the same across a wild number of species. So I said, well, we know how to do it for organismal phenotypes. Can we do it for gene expression? But instead of studies where you do 5 or 10 phenotypes, traits, such as height, weight, whatever, we have tens of thousands of genes across the genome. Can we do this experiment? And so we did that. What we did is, several years ago, we grew up 200 land races of rice and 20 advanced breeding lines in both well-watered and drought environments in the International Rice Research Institute. And during the course of that experiment, we measured everything we possibly could, but the ones here that are larger are the ones that are important for this talk. We did 1,320 RNA-seq reactions at one point in time, basically. So that's one of the weaknesses. We only have one time point, but obviously you can't do that for multiple time points. It's too expensive, but we decided to start with one time point that made sense. And we also counted fitness by looking at grain number or mortality. The way we were able to do 1,300 RNA-seq reactions is, this was Rahul Satija of the New York Genome Center, who's a colleague of mine at NYU. We modified his drop-seq protocol. So part of the drop-seq protocol is the STAMP protocol, which is a single-cell protocol for single-cell transcriptomics protocol. We just kind of like took that and used it for our purposes to use the barcoding rather than the single-cell feature of that. But it worked well. We were able to do 1,300 libraries in 3 days and cost us about $6,000 to do. So we were able to do this rather quickly. And we have expression, essentially after we filtered everything, we have expression about 15,000 genes across our entire sample, a fairly large number. And these are just the genes that are heritable. Actually, all of the transcripts we have are heritable. That is, there's a significant genetic component to their variation. We also counted seed, every seed on each of those plants. We had 4 million seed. Counted in the Philippines, in the International Rice Research Institute. So we're able to tell you whether this land raises a lot of seed or not a lot of seed. And of course, as I said, now we have RNA-seq. So now we can do this experiment, right? We can look at fitness and we can look at levels of gene expression, where it's so easy. Nobody had done this experiment in this large a scale, with 15,000 clearly confounded correlated characters across. And so we're not really at this point able to take full use of this data set, but we treated everything as just individual independent terms. Instead of measuring the selection gradient, we measured what's called S, the selection differential. And the selection differential, the difference between beta measures the total direct and indirect selection. That is, if fitness is the trait, if height is the trait I'm interested in, increasing height may have a direct effect on fitness, but it may also have an indirect effect. Maybe increasing height also increases root biomass, which has an effect on fitness. So beta is able to measure both direct and indirect. Beta is only able to measure the direct component of fitness. S, the selection differential, puts those two things together, both the uncorrelated and correlated parts. It turns out that beta and S are actually strongly correlated with each other, so I think in terms of what we learn, it's the same. So what do we have? We now have, for these 15,000 genes, a distribution of selection strands for expression at this one time point. And what do we find? Essentially, it's very similar to what was found in normal organismal phenotypes. It is somewhat, most of the strength of selection is neutral or nearly neutral. That is, there's not an effect. There is a longish tail, but the tail is not as long as you see in organismal phenotypes. Essentially, above a selection differential of about 0.2, I think there's only less than 1% of the transcripts have a selection differential less than that amount. Under drought stress, we see a shift in the distribution of the selection differentials. That is, there's greater strength of selection under drought stress. So levels of gene expression matter more when you're under drought stress than when you are under normal, well-watered conditions. And as I said, they behave similar to what we see in organismal phenotypes. Not in the normal conditions. In the normal conditions, everything lived, so there was no mortality. But in the drought stress conditions, some plants died. And so we're able to look at two different types of fitness measurements: fecundity, which is the number of seed, and survival, which is whether they lived or died. And in general, again, the distributions are hewing close to S equals 0. But now we're seeing that there's also a shift. If survival is your fitness component, what we're seeing is that there's strong component of negative selection. That is, there is selection for a lot of genes to be repressed in their expression in order to have high survival. And that's how we're seeing that. And actually, this has been seen in not quite these traits, but in organismal phenotypes, the one that Hopi Hoekstra and Joel Kingsolver have done. They also see a difference, these types of differences in the distribution of selection strengths when you're looking at different fitness components, such as fecundity or survival. Some of the things that are coming out, what we find is half the genes in the genome in terms of their expression, have either what we call conditional neutrality or antagonistic pleiotropy. That is, half of the genes in the genome, in terms of their expression level, are either neutral, S equals 0 in one condition, but highly positive or negative in another condition, that is drought versus well-watered, or they switch signs. In one condition, they have negative selection. In another condition, they have positive selection. It's about 52% of the genes have that behavior. And that's important because in classical evolutionary theory, we think that conditional neutrality and antagonistic pleiotropy have a strong role to play in local adaptation. We look at different factors that affect the strength of selection. What factors in the gene or that gene is affect? And there's a lot of it that affects it. One is expression level and expression noise. It turns out that the higher the expression level of the gene, the lower the selection strength. In other words, if you're a gene that's highly expressed, changing the level of gene expression is not affecting fitness as much. Which kind of makes sense. These are probably the big constitutive genes that are always on and always at high level, so there's not much movement in terms of its effect on fitness. Interestingly, expression noise is the same thing. Very noisy genes, that is genes that are bouncing around in expression among different individuals in the same environment. So it's got a lot of noise in the expression, actually also have very low selection strengths. Again, that kind of makes sense, right? Because if you're a gene that somehow fluctuates stochastically in one environment, you don't want that stochastic noise to be fluctuating your fitness a lot. So you're keeping selection. And we're seeing other things that are associated with that. One of the things that's interesting is we look at connectivity in a gene network and different levels of connectivity. These are the three different experiments: fecundity, survival in drought and in well-watered conditions. And it shows the same thing. Genes that have high connectivity have low selection differentials. Very hub genes that are the center of networks, their selection on gene expression is very low. In other words, if you try to tweak their expression a little bit, it doesn't have as much an effect on fitness. It's not a very big change, but it's actually a significant change, and it's seen across the different levels. And then this other one was kind of interesting. It's significant only because we have a large data set. But genes that are found in the disposable part of the pangenome. Remember, the rice genome, like other crop genomes, has a pangenome. Not every landrace has all the genes that other landraces have or other individuals have. There's a core set of genes that everybody shares and a dispensable part of the genome that only some landraces have but others don't have. If you are a gene in the core set, you have essentially lower selection differential. A lot of the genes that have a high selection differential for gene expression are in the dispensable part of the genome. And again, that's significant across these different classes. And we can do things like, of course, look at what classes of genes or gene groups are selected in a variety of different conditions. I'm not going to go through all of this with you. Finally, what I'd like to show is that the one thing that's interesting is now that we have the distribution of selection differentials, which genes have such strong selection that they're significant, okay? And why is this interesting? Especially in the case of drought, which this is. These are genes that might be interesting for manipulation in breeding or in genetic engineering in order to deliver higher fitness or higher yield. And what we find is that we have several uncharacterized proteins, but if you look at the right side under positive selection, for fecundity, what we're seeing is that at least two genes that are part of the photosynthetic machinery, and a nitrite reductase, it's part of the chloroplast nitrogen metabolism, there's strong positive selection. That is, increasing levels of expression of these genes will increase the grain number or the seed number under drought stress. And for survival, there are two genes, a hypothetical gene and a MADS gene, OS-MADS18. Again, increased levels of OS-MADS18 will increase survival of the plant. And this just shows you, this is a graph of selection coefficients across the different genes in the genome. Look at OS-MADS18. It's way up there. It's by itself. Its S is 0.77, very significant. And what is it? It's a gene that's a major QTL for flowering time, and it's been shown to be major QTL for fitness under stress. What it does is that expression of this gene promotes early flowering. So what is this gene? It's a drought escape gene. So under drought stress, those plants that flower earlier are going to survive. And so what this gene does is those that have higher levels of expression and therefore early flowering are able to escape the drought. And this just shows you essentially the selection analysis on the trait itself, not on the gene expression. Flowering time, relative fitness, you can see the relationship. Early flowering gives you higher fitness. So in this part of the work in the lab that's been going on now for a few years, we think we've managed to find a way to try to directly measure the strength of selection or gene expression in the genome. We've only done it for 2 conditions in a set of plants and in 1 time point. We would like to now look, we're now going into 2 directions here. One is we would like to see what's happening in other tissues of the plant. Is the strength of selection concordant across the different tissues? Or if this gene is expressed in, say, flowers and leaves, the strength of selection is antagonistic? I mean, we don't know those details. It would be interesting to find out, because this tells us something about the nature of selection on this level of phenotype. And the other is we're taking those genes that we found and we're making transgenic lines to see, to put our money where our mouth is. We think these are genes that are important for fitness of the plant under drought stress. Can we show that? And can we show that by making transgenic plants? So we've just been given a grant by a private foundation to do that. So those are the 2 projects that I've told you about. I think I may have gone really fast, but I hope you got kind of those 2 messages. We now have a map of fitness consequence in the rice genome. We're able to tell you across the rice genome what we think the level of selection is, even in non-coding areas. We need to continue to refine this by adding more data. I think we have a first draft of it, and hopefully this will become useful to those of us who are genomicists, those of us who are looking at molecular genetics, and those of us maybe who are interested in genetic mapping. And the other is we now have a way to measure the strength of selection on gene expression, something that's never been done. And now we can try to understand the linkage between levels of expression of a gene and the strength and its usefulness for adaptation and fitness. So this work was largely done by, I have 2 labs, one in New York City and one in Abu Dhabi. NYU has 2 we had 3 campuses actually, one in Shanghai. But having three labs would be too much. So 2 labs, I have one in New York and Abu Dhabi, and we did this project mostly in New York with some help from the Abu Dhabi portion of my lab. These are the people who are involved. The ones in asterisk on the left are the people in the lab who have been really involved in these 2 projects and are collaborators on these projects on the right. The work that I told you was largely funded by the Ziegler Family Foundation and also with some funds from NSF, Plant Genome, the Moore Foundation, and NSERC in Canada. And with that, I thank you, and we have time for a few questions. So, any question? There's somebody over there. Yes.
Was the drought conditions, would you expect plants that escape drought to do well in that drought? Was it an early or a late drought? Was it strong?
We did this at the International Rice Research Institute, and they imposed an intermittent drought condition throughout the entire life cycle until about, for the first 2 months. So it was intermittent. It was meant to stress the plant without necessarily killing them, because we wanted to, of course, make them survive. So it was an intermittent drought condition. So, any questions? Yes.
I have another question about the drought stress experiment. So, sort of rule number 1 about screening for genetic components of drought tolerance is you separate the plants into those that flower at different times, so you don't accidentally map flowering time.
Yes.
So I'm wondering if you're able to separate your population into those that flower early and flower late, then were you able to see things that are more specific to the drought per se rather than flowering time?
Right. So that's a really good question. We did stratify. There was clearly some very late flowering lines. We removed them from the analysis because it was going to skew our data. And so we have a normal analysis around flowering time. We have tried to look to stratify it according to similar flowering times. The problem is we begin to lose power dramatically in this experiment. We thought 200 was a large experiment. Clearly, to do this, we need several thousand. But you're right, and we've kept that in the back of our mind, and that's correct about that.
Thanks, Michael. That was brilliant. So I was really surprised by the Rubisco small subunit coming up and so strong. And of course, you have the Rubisco large subunit that's in the chloroplast. So I guess a couple of questions is, was the chloroplast part of the fitness map, the chloroplast genome? And then that coordinated expression has got to be a interesting piece in the puzzle.
Yeah, so we haven't dug, okay, there were several things. We don't see the same strength in the large component of Rubisco. We do see that other components of the photosynthetic machinery have relatively high selection differentials, but not so high that it's significant. These were Bonferroni corrected. So they were pretty high, but they didn't pass our Bonferroni threshold, so we don't talk about that. But it's clear that photosynthesis is important. But it's an interesting question because now we're trying to figure out the coordination of the small and large subunits, and we need to dig into our data to see to what extent we're seeing across our lines a coordination. But it's clearly something's going on with photosynthesis there. That's important. Anything else?
Address the question of, so in vertebrate genomics, it's quite easy to move from human to mouse to chimp and generate reasonably useful conservation scores.
Right.
In vertebrate genome,
Yeah.
that shows any.
Yeah.
Yeah. In vertebrate genomics, it's reasonably easy to move from people to primates to alligators and whatever else and generate conservation scores that are informative in non-coding regions. But can you say something about why that doesn't work in the plant world?
I have no idea why this is work on plants, but you're right. And so, in fact, when we started to do this, we're only able to do our FastCon scores, for example, only like within ERISA in different levels. I have no, you know, I don't know. I have to admit that I used to stay away from the conserved non-coding sequence area of work because I just didn't know what to make of it. Now we're having to go back into it and see what that means. But I don't know.